Whenever we have a set of bacteria of the same species that are isolated, and just for an example, group A strep, the strep throat germ, the group A streptococcus, those bacteria exist as a population. So there are some members of that are identical that may belong to the same family. Some will be related by tribes, and some are only distantly related. So one of the challenges that we have from time to time is we may have a cluster of isolates in the community or in a hospital, and the question is, are they really related or not? They're the same species, but are they really the same? So we then have to resort to another method of typing, and there are a number of these that we kind of group together under the heading of DNA fingerprinting of bacteria. So basically, we start with a group of bacterial cultures that have been identified as being the same species. We can then take the DNA from those bacteria, run it through a PCR type of reaction that then generates a set of fragments of DNA from those bacteria. We then can run it through another machine that essentially sorts those fragments by size into something that looks a lot like a barcode. And then by that sort of looking at the different barcodes, you can compare them, line them up side by side and say, are these alike or not? If we didn't have this capability, it would make our infection control efforts, number one, much more difficult. We would have to treat any cluster of isolates the same as a possible outbreak and immediately go and expend the resources and often chase down a lot of dead ends, because many of the times when you have a cluster, it's not really an outbreak. So what this does is allows you to most effectively target the resources you have, so you're not pulling the trigger on an investigation when you don't need to, but you know you really need to be doing it well when you do have a cluster.